Okay, so we will begin with case study 1. For our first referral, we have Jocelyn at age 41, and she has come to us to have some karyotyping done because she is 17 weeks pregnant, and her age puts her at a higher risk of having a child with certain birth defects. Here is the karyotype that we were given of Jocelyn's baby's chromosomes for this case study. When looking over her baby's chromosomes, we can see that there's a translocation mutation between chromosomes number 14 and 21. Specifically, this is called a Robertsonian translocation since it is involving the 14th and the 21st chromosome. This mutation is likely to cause the patient's child to have Down syndrome should she continue on with the pregnancy. So, what does this all mean? The life expectancy of children with Down syndrome is variable. Some have been known to live full, healthy, happy lives, while others don't quite make it past their teens. It all depends on how severe their symptoms are and how they are being managed, or if they even can be managed. So there are multiple support groups that are available for those dealing with Down syndrome. They are the National Down Syndrome Congress, NDSC, and the National Down Syndrome Society, NDSS. For the second part of the human genetics case study, we were given a patient's genome sequence and had to translate another from an electropherogram. Our patient is a 27-year-old male who has recently moved from an area of low elevation to high elevation. He says that he has been experiencing inflammation and severe joint pain. We were given his genome sequence to find out why this could be. First, we took the given sequence and the sequence from the electropherogram and translated to the complement and the mRNA sequence. Using the mRNA sequence, we were able to convert the codon to protein. And when comparing the patient's sequence to the sequence from the electropherogram, we can see that a missense mutation occurs, causing the fourth protein in the sequence to be changed. A missense mutation occurs when a singular nucleotide change causes the codon to code for a different protein. In this case, the GAG codon was mutated into the GUG codon. And this type of mutation will not necessarily cause a disease, but it will cause a malfunctioning protein. For the third week of case studies, we were assigned a fly protein sequence, and we used FlyBase to help determine the related human disease. When putting the sequence into FlyBase, I found that the best hitting gene for the sequence is CG3376. This gene is found to be responsible for catabolic reactions as long as biosynthetic processes. Therefore, it is thought that the loss of this gene would result in either the loss or deceleration of catabolic reactions. The human disease that was identified for the protein sequence is Niemann-Pick disease. This disease occurs due to a buildup of the lipid sphingomyelin in liposomes.